Okay, this is a screencast for Bio 234 genetics, dealing with population genetics, which is the next section in the syllabus. So the things that hopefully you will be able to do and know by the end of this section is know what the conditions are that are required for using Hardy-Weinberg equilibrium equations as an explanatory tool. You should be able to calculate genotype frequencies from allele frequencies within populations. Using Hardy-Weinberg equilibrium equation, you should know the four forces of evolutionary change that alter population allele frequencies over time. One definition of evolution is basically just that allele frequencies are changing over time. Describe the 5 assumptions that must hold for Hardy-Weinberg equilibrium to hold. Know how to calculate the probability of fixation for a population and how this applies to the persistence of rare alleles in populations, and then know how to project evolutionary change in a population in population structure due to selection in populations. So that sounds like a lot, but it's all highly interrelated. So hopefully the concepts will easily be able to be related to one another. So when you think about genes, you can think about genes as units of inheritance. That's what we just got finished doing in class when we were talking about how genes get passed from one generation to another. You can think of genes as a DNA sequence, which is what we talked about in the first half of the class, talking about how that DNA sequence gets transcribed into messenger RNA and then translated into a gene. And then we can also look at genes as characteristics within a population and look at the frequencies of alleles of genes and how they change as you move from one position on the Earth to another position on the Earth, moving from population to population. So when we think about a population, we can think about a population of alleles or haplotypes. And so here we have essentially 100 different alleles. 70% of them are the blue allele and 30% of them are the yellow allele. And so we could say that the allele frequency of the blue allele is 0.7 and the allele frequency of the yellow allele is 0.3. And we can estimate these by basically sampling populations and doing sequencing on them, looking at microarrays, looking at complementary DNA on microarrays and things like this. But essentially, this becomes one of the fundamental units, this allele frequency, and we're going to be talking a lot about that in this chapter. When we think about alleles in a population, we can think about these alleles being partitioned into individuals, and because we oftentimes talk about diploid species, because a lot of the species that we think about on a regular basis are diploid, we can think of these genotypes or these allele frequencies as being organized into genotypes. And so on the left-hand side here, we have allele frequencies, and on the right-hand side, we have genotype frequencies. And if we have 70 out of 100 dominant alleles, the P is 0.7, we can calculate using Hardy-Weinberg equilibrium equation, which is down here in genotype frequency land. We can basically say that P squared 0.7 is 0.49. 0.7 squared is equal to 0.49. So we should expect 49% of the genotypes from this population of alleles to be homozygous dominant. 2pq is the frequency of heterozygotes, and q squared is the frequency of homozygous recessives. And we do this because there are two ways of having heterozygotes. That's why it's 2pq. This is essentially the Hardy-Weinberg equilibrium. And one of the things that we know about Hardy-Weinberg equilibrium is that if you don't have anything unusual going on in your population, these allele frequencies won't change over time. And as a result, the genotype frequencies won't change over time. So from generation to generation, you end up with the same allele frequencies over and over and over again. But that only happens if you make certain assumptions. And you guys have talked about this in lab, and I do use slightly different language than Dr. Reynolds does when I talk about this, but we're basically both saying the same thing. One of these assumptions is that mating is random, and so the assumption is that mating occurs at random. If there's non-random or assortative mating, then genotype frequencies and allele frequencies will be altered. No mutation, no migration, no movement of individuals into or out of a population. Population sizes are extremely large, meaning that there's no genetic drift, and there's no differential reproduction, and this basically means that there's no selection going on. And Dr. Reynolds talked about this as being a situation with infinite resources, but you have to consider that that would imply that all selection is being imposed by resource-limited competition, but there are all sorts of things that can contribute to selection. Climatic factors can contribute to selection. There can be sexual selection, behavioral selection that doesn't really have anything to do with underlying resources. So basically, we have the assumptions in this table. We have what happens if there's a violation to these assumptions, and then what the impact of that is on the population. So in mutation, where mutation is concerned, if there is no mutation, allele frequencies don't change, but mutation, by definition, brings new alleles into the population. It's how new alleles arise, and then that changes the frequency, even though minutely, of the alleles that were already there when you add a new allele to the mix. So genetic drift is a situation where allele frequencies are changing at random, and selection is where allele frequencies depend upon selection pushing allele frequency change in a particular direction. And so evolutionary change, once again, is just a change in allele frequencies over time. And so any of these can lead to evolutionary change, although not all of them are going to lead to directional evolutionary change. So we can think of these as something happening to population in generation one, and then happening to it under new scenarios. So we can take individuals and we can have a mutation in one of the individuals that gives us a new allele. Although the overall allele frequency hasn't changed a lot, it has changed minutely as a result of that. Population 1 can actually have some individuals added to it, making it a larger population. Depending upon the mixture of alleles in that new addition to the original population, you might have a different allele frequency in the resulting combination of those two populations. If population goes through a genetic bottleneck, a population size bottleneck, where you go from a lot of individuals to relatively few individuals, and then that population then gives rise to another generation that is larger, that larger population can bear the mark of any random changes in the downsizing of that population from generation 1 to generation 2, such that then you have different allele frequencies here that then get perpetuated in the next generation. And then you can have selection occurring such that in this case, it looks like it's the blue allele that is becoming more and more selected for from one generation to another generation, because the blue alleles leave more offspring to the next generation. And so this basically is mutation, migration, genetic drift, and selection, which are 4 of the 5 violations of Hardy-Weinberg equilibrium. Non-random mating is the 5th, and it was the first one that was listed in that table, but it's the first one that we'll go into in depth. And this is a situation where allele frequencies are changed because either like individuals prefer like individuals. Homozygotes tend to increase, heterozygotes will decrease. So homozygous dominants will mate with homozygous recessives, and homozygous dominants will mate with other homozygous dominants. Homozygous recessives will mate with other homozygous recessives, and so you get a preponderance of homozygosity in the population, altering genotype frequencies. But in this case, it doesn't alter allele frequencies. It just changes the distribution of those allele frequencies among the different genotypes. In a situation where opposites attract, you would have homozygotes decreasing and heterozygotes increasing, because you're getting more breeding of homozygous dominants with homozygous recessives right there, producing a lot of heterozygotes. Once again, allele frequencies are not changing, but genotype frequencies are changing. And as a result, Hardy-Weinberg equilibrium equations don't predict adequately what the genotype frequencies are going to be. We can calculate what the likelihood is that you possess a particular allele because you inherited it from a common ancestor if there's inbreeding. So inbreeding can also mess up allele and genotype frequencies. And so here's a situation where we have two great-grandparents who gave rise to children who then gave rise to children, and then those children mated with one another, such that there's a certain probability that this allele possessed by this individual at the bottom of this pedigree has a mutation, not because they got it from one of the outcrossed individuals here in the middle, but because they actually got it from their great-grandparents. So this would be their parents, their grandparents, their great-grandparents, the great-grandparent male up here. And we can calculate this by basically, if it's a two-allele system, what are the chances of inheriting this allele from the parents? It's half for each leg in the pedigree. So from here to here, your probability of inheriting that allele is a half. From here to here, it's a half. From here to here, it's a half to the great-grandfather. From here to here, it's a half. From here to here, it's a half. And from here to here, it's a half. But then you also have to consider that there's the grandmother up here. And so there's also the probability that you could have gotten it here to here to there, to the grandmother, and then through her offspring as well. And so you would add both of those paths together. And so that's how you basically calculate the probability of identical, two alleles being identical by descent, is by looking at all of the shared common ancestor pathways and then adding those cumulative probabilities together. So we've already talked about mutations affecting allele frequencies. Generally, mutations don't have much of an impact on populations because they start out at relatively low frequencies, and the probability of fixation, the probability of an allele going from just being represented by one copy to being found in every individual in the population, the probability of fixation is equal to the allele frequency at the beginning. And so if you have 100 individuals in your population, well, you have 50 individuals in your population, you have 100 alleles in the population, one of those is mutated so that now you have a frequency of 0.011, is that right? Yeah, 0.011% of the population, then that is the probability that it will become fixed. And so the only way that you really get fixation of rare alleles like this is if they have some selective advantage. And so mutation rate is usually too low to affect populations unless another factor is at work. And it could be any of these, but it's probably going to be differential fertility. It could be small population size. It could be something like migration, but oftentimes we think of these things spreading rapidly through populations due to natural selection. So we can look at a complicated pedigree like this, and we can ask ourselves, what's the probability that one of these offspring has a particular allele, not because of new mutation, but because this individual got it from some shared ancestor in the past. And the way we would have to do this is we would have to say, well, the probability of this individual getting it from its parent is half. Probability of getting it from their parents is half. The probability from getting it their parents is half. And then that would take us, would that take us? Then we have to go all the way up here to the top and then come down the other side of this pedigree and calculate each of those probabilities. And so anytime you have what's known as consanguinity, when you have individuals who are marrying close relatives, there's the possibility that you get the homozygous condition not because of anything other than the fact that you have inherited it from your ancestors who are all related to one another. And so these individuals have lots of consanguinity going on in this particular pedigree, and so you can basically get more homozygous recessives coming into this situation because of inbreeding. This is how we get the accrual of deleterious homozygous recessive traits accruing in certain populations. We saw this back when we looked at hemophilia. You have a certain amount of inbreeding within the royal family, and that has led to a higher than expected rate of hemophilia in the population. So mutation with non-random mating, one mutation occurs, and then that mutation tends to become propagated in the offspring because you have a lot of inbreeding in this particular pedigree. Migration can affect allele frequencies, and there's two forms of migration here. In this case, migration is prevented, so the population gets divided into two segments, two subpopulations, and there is no movement between these two populations in this top panel here. And whatever the subpopulation's allele frequency is, that's the new allele frequency for these two subpopulations. And as long as you don't have gene flow moving back and forth, they will once again, under Hardy-Weinberg equilibrium, maintain the allele frequencies of the subpopulation structure that is caused by the intervening of some barrier to dispersal. The other thing that can happen is allele frequencies can change when migration is allowed, such that we have the original population, we add 20 individuals into it, but the number of individuals that are there have a different frequency of alleles, such that then that affects the final allele frequency. Because you have fewer of the blue allele in the 20 individuals migrating into the larger population, that reduces the overall frequency of the blue allele a little bit and increases the frequency of the yellow allele a little bit. And so the degree to which migration changes the population genetic structure of the population is dependent upon the size of the difference between populations and the number of migrants compared to the original population that's being added to. And so if you have a really big population and a small number of migrants, even if that new number of migrants has a very different allele frequency, it's not going to have that big of an effect because it's just a drop in the genetic bucket, so to speak. Genetic drift also changes allele frequencies, but it does this in a random sort of way. And so if you were to take a random sample of this population that is much smaller, that allele frequency may be very different from the original population, simply because you're taking a non-representative sample, because you're drawing out genotypes out at random. And so you might see a situation where it goes from P of 0.7 and a Q of 0.3 to P of 0.8 and Q of 0.2, and then that then serves as the population, serves as a basis for that population expanding. This is what happens in population bottlenecks, and this is from conservation biology a very important thing, because when we think about populations that are under stress from hunting, for example, you might expect that population to get very small. And then when you come back and undergo conservation activities, you then create a much larger population, but that larger population is stuck with whatever the allele frequencies and diversity of alleles that was found in that much smaller population. So even though you have more individuals, those more individuals are now genetically less diverse than the original population. This is a situation that's happening in cheetahs. Presumably cheetahs have undergone some population constriction, some bottleneck in the past. And so one of the problems with cheetahs is that they're all very similar to one another genetically, because they have presumably gone through one of these genetic drift sorts of events that basically gives you a smaller selection of the original genotypes that alters allele frequencies. And then those allele frequencies get expanded up into current numbers of cheetahs, and there's just not a lot of genetic diversity there. Drift can have a large impact. So this is basically a bunch of populations that are being simulated. You start off with an allele frequency of P is equal to 0.5 and Q is equal to 0.5, and then it just simulates change due to drift if the population size was only 100 individuals. And you see that some populations go to fixation, the blue and the purple at the top, and you might have some situations where P goes down to 0. It hasn't happened in this case, but you could also lose an allele in a population, not from any selection acting, just a bunch of random fluctuations due to population size. If you lowered that population size to a population of 10, you would find these populations going to fixation or going to extinction of the allele in much shorter periods of time. And in class, I'll show you a program that you can use to do these kinds of simulations. So the founder effect is one example of this, when a small population expands to a larger population that's isolated geographically or whatever from the surrounding populations. So founder effect is when you go through one of these population bottlenecks because a group of individuals migrates away from a large population, a small group of them go off and found some new population that is no longer connected to the original population. And so when we think about human beings moving around the earth, that's ultimately what has led to a lot of the population structure of human beings. Different subpopulations of humans have different allele frequencies of the wide variety of different alleles. This is also what happens when organisms migrate out onto oceanic islands. You have a small number of founders. You only have the alleles that they possess founding that thing, and then there's no connection back to the original population that allows you to have gene exchange, allele exchange, to kind of equalize those allele frequencies. And then the last thing here is the effect of selection on allele frequency. And so if you have directional selection, what it means is, in this case, that individuals that are homozygous dominant leave more offspring than individuals who are homozygous recessive. And over time, generation to generation, the allele frequencies are changing because you're getting a preponderance of dominant alleles in the population and a predominance of homozygous dominant individuals. Now, this is a simulation that your book has provided where there's directional selection such that the big A allele is favored, but we can also go through a mathematical way of figuring out what these genotype frequencies would be under selection. And so we begin with just the Hardy-Weinberg equilibrium equation, which is p squared plus 2pq plus q squared. And that would normally equal 100% of the population, 1.00. But if we then have fitness consequences, W's are fitnesses, and these are all relative fitnesses, such that we have the fitness of the homozygous dominant, the fitness of the heterozygote, and the fitness of the homozygous recessive, multiplied times the number of those individuals possessing those genotypes. That then gives us how many of these genotypes, how many of the individuals possessing these genotypes we would expect to find in the next population based on what their fitness of those different genotypes is. And so in one case, we might assume that anybody possessing a dominant allele has a fitness of 1, and anybody possessing 2 recessive alleles are under selection. So this is essentially selection against homozygous recessives. Homozygous dominants and heterozygotes have the same fitness, but homozygous recessives have a lower fitness. The problem with this is that this won't equal 1 because you're going to have fewer homozygous recessives in the next generation. And so the question becomes, well, how do we then correct for that? Well, what we can do is we can calculate what the average fitness is of this population and then divide each of these units, p squared times 1, 2pq times 1, and q squared times 1 minus s. We can take each of those and divide them by the average fitness. And the way we calculate the average fitness is essentially by averaging up what all of the fitnesses are across all of these different genotypes. There's an understood 1 for p squared and 2p cubed, but there's 1 minus s multiplied times q squared. And as it turns out, the average fitness equals 1 minus s q squared. And in class on Wednesday, we will go through the derivation of how you get that. You might want to try and work it out yourself, though, between now and then. Once we have that average fitness, we can divide the fitness of each of the genotypes by the average fitness, and then those will all end up equaling 1. And at this point, we're going to stop there because I want to go through this math with you in class so that you fully understand what all that is about. If you want to work this out, you end up having to convert all of the p's to being expressed as q. And if you do that, then you can actually solve for q squared, and it will give you this answer. And we'll work the math in class. All right.